Today's video is about the first life forms. A remote and desolate corner of Australia was nicknamed the North Pole by disappointed gold prospectors in the 1800s. But this region has been a gold mine, after all, for 20th century scientists. It was there that the oldest known cellular fossils, 3.5 billion year old traces of early unicellular organisms, were found. So these are a couple of examples of the oldest organisms that have been found so far. They are fossils that are 3.5 billion years old. Scientists look at the DNA RNA protein relationship to try to figure out where heredity started. Why is RNA necessary? Why can't DNA do protein synthesis itself? The answer is in RNA's shape. It can take many shapes. Remember, we have mRNA, tRNA, and rRNA. These shapes are made by hydrogen bonds. So some RNA molecules might act like enzymes and catalyze chemical reactions. In the early 1980s, Thomas Ketch found that a type of RNA found in some unicellular eukaryotes acts as an enzyme. He named this ribozyme. And there it is. So sometimes some scientists believe that life may have started with self-replicated RNA that would have genetic information. It also could evolve. Can't listen to that because I have no speakers. Later studies showed that ribozymes can act as catalysts for their own replication. Self-replicating systems of RNA have been created in the lab. This supports the hypothesis that life could have started this way. Self-replicating RNA is not only the first case of heredity, but also the first case of competition. So think back to what I just talked about in the last video about microspheres and coacervates basically being cell membranes that are able to grow and bud and reproduce. Now we have self-replicating RNA. So if you combine hereditary information with cell membrane, you start to get a picture of how cells were first formed. Because RNA can have many forms, these would have competed for available nucleotides. Whichever type 1 would survive. Now we've discovered other ribozymal activities. RNA plays a role in DNA replication, protein synthesis, RNA processing, and other biochemical reactions. There are different hypotheses about how RNA could have evolved into modern cellular life. There are certain kinds of minerals that have formed a template on which organic molecules could line up and form polymers, which we know are the basis of the molecules of life: carbohydrates, proteins, lipids, nucleic acids. Or self-replicating RNA could have started to evolve inside cell-like structures like microspheres and coacervates. Here, RNA would have provided hereditary information that these structures lacked. If RNA was able to direct the assembly of structures like microspheres or coacervates, a cell-like system would be formed. So like I said, the last video we talked about how the atmospheric conditions allowed for certain organic molecules to start to be able to form, which led to the formation of these cell-like structures, microspheres and coacervates. Now we have a self-replicating RNA which can conduct different processes. Now, there's no direct evidence that exists as to how the first cells evolved, but we can make some inferences. So let's go ahead and look at this. So the evidence: when first life began, there was little or no oxygen. So the inference from that is that the first cells must have been anaerobic. You cannot be an aerobic organism and survive without oxygen. That's just not possible. So we have evidence and we make an inference from it. Next piece of evidence: the oldest fossils are thought to be cells similar in size and shape of some living prokaryotes. So the inference from that is that the first life forms were probably prokaryotic. Another piece of evidence, the first cells might have developed in organic rich environment. The inference there is that they were heterotrophic. Putting all of this together means that the first life forms were probably anaerobic, heterotrophic prokaryotes. So, a growing population of heterotrophs, depending on spontaneously formed organic molecules for food, would eventually remove most of those organic molecules from the environment. So now autotrophs would start to have an advantage because the organic molecules are being used up by the heterotrophs. But the first autotrophs were not photosynthetic. They relied on a process called chemosynthesis. Archaebacteria are unicellular organisms that live in harsh environments. Many are autotrophs that get their energy through chemosynthesis, which is basically photosynthesis without the light, and instead they use inorganic molecules. By about 3 billion years ago, some forms of life became photosynthetic. Scientists infer this from geologic evidence, chemical traces of photosynthetic activity. The most oldest known, that is some terrible grammar, most oldest known, holy crap, sorry. The oldest known fossils are similar to our modern cyanobacteria, which you see here. Cyanobacteria are photosynthetic unicellular prokaryotes. However you pronounce that is a genus of modern cyanobacteria, Limbigia. Let's go with that, Limbigia. They grow in colonies and form layered structures called stromatolites, which are those mound-looking things that you see there. The oldest known stromatolites are 3.5 billion years old. Okay, so we know that oxygen is a product of photosynthesis. So I want you to think, like, let's go back and think about what kind of environment we're talking about. So the first life forms, there's no oxygen in the atmosphere, so they have to be anaerobic. They're heterotrophs because there's lots of organic compounds around, but they start to use up all of the organic compounds. So then you step in the autotrophs, and autotrophs can make their own food and they produce oxygen. The first autotrophs were chemosynthetic, but they would still start to produce oxygen. And then you would have the photosynthetic organisms like the cyanobacteria, which would evolve shortly after that. So oxygen being a part of photosynthesis means that we can start to lead up to respiration. But for a lot of early organisms, oxygen was actually damaging. For others, oxygen bonded to other compounds. It did not damage them. These were the first steps in going towards aerobic respiration because you can't have respiration without oxygen. It took 1 billion years for oxygen gas levels to reach today's level. Eventually, they reached the upper atmosphere where it was hit by sunlight. Some wavelengths of sunlight react with and split oxygen into a single oxygen atom, and it reacts with O2 to form ozone. Ozone is actually poisonous to plant and animal life, but it protects from the UV radiation, which is why it's only in the upper atmosphere. So now we have both heterotrophic and autotrophic prokaryotes. So how do we get to the first eukaryote? So we go all the way back to the theory of endosymbiosis, which is a successful mutually beneficial relationship between a large prokaryote and a small prokaryote. Remember, I talked about this when we studied the mitochondria and the chloroplast. The chloroplast is what does photosynthesis. It is a double-membraned organelle that contains its own DNA, and only chloroplasts can make new chloroplasts. Same thing with mitochondria. Is the only double-membraned organelle that contains its own DNA. And it's interesting that the chloroplasts are responsible for photosynthesis, which creates the food, and the mitochondria uses that food to create energy. This is where the theory of endosymbiosis comes from. So scientists infer that the endosymbiotic aerobic prokaryotes evolved into modern mitochondria because of aerobic respiration. Later on, photosynthetic cyanobacteria may have evolved into chloroplasts. Of course, we need evidence for that. Both chloroplasts and mitochondria replicate independently from the cell cycle. They also have their own genetic material that differs from the rest of the cell. The DNA of a mitochondria and chloroplast is in a circular arrangement, just like prokaryotes. Bruh. So that is the end of chapter 14. Don't forget, we have a Zoom meeting on Friday morning. If you want to attend, please come prepared with questions you would like to have answered, either for biology or chemistry. And if it's just really super boring and we're all just sitting around staring at each other, I will do a review.